previous lectures, we have covered topics including microscopy, radioisotope technique, chromatography techniques and electrophoresis. All of these techniques are widely used for different applications in the various fields of biotechnology. In this lecture, we are going to start a new technique, that is centrifugation techniques. Now, centrifugation is one of the most important and widely applied research technique in biochemistry, in cell and molecular biology, in medicine and in various other areas of biotechnology. Our current research and clinical applications rely on isolation of cells, subcellular organelles or fractions, membrane fractions, macromolecules and lot of other constituents of cell in sometimes in high yields. Now, this separation technique exploits the inherent varied sedimentation property or sedimenting property of substances for their isolation by the application of centrifugation or centrifugal field. It is used as to separate or concentrate material suspended in a liquid medium, and the resulting solution has two components, namely the sediment and supernatant. Now, before we go into the details, let me give you an overview of this technique and let us understand little bit about what we are going to discuss in this particular lecture or in this particular section. All right. So, what we were like first thing is, let us understand this through a very simple experiment. Now, if I take say certain particles of a particular size which are suspended in say liquid medium, what is going to happen to these particles which are of a certain size? Like after some time, what is going to happen is, after certain period of time, these particles will settle down at the bottom. Now, these particles settle down, they will certainly take more time to settle down, but these particles settle down because of gravitational pull or gravitational force. Now, if we take an example here, that if the size of the particles is larger than these particles, then they will take less time, that is gravitational pull will be more and they will settle down faster. Supposing there are particles which are smaller than these particles, then they are going to take more time to settle down. So, what we see is the size plays an important role, where through gravity a particular particle is settling down. Now, the rate at which they are settling down is depends on, like we have seen, size, but also the gravitational force actually, or the pull, gravitational pull. Now, if you want to increase the rate of settling or sedimentation, we can call it, then you have to increase the force on these particles, so that they can settle down faster. And this force could be given by given in the a particular centrifuge, that is, when you rotate a particular tube containing this material or these particles suspended in a liquid, then they will experience a particular centrifugal field and they will settle down faster. Now, remember, as the particle size goes, now there are certain factors on which this whole thing depends. One is, like we are talking about, size of the particle. Another thing would be shape of the particle. Supposing you have a spherical material of a particular molecular mass and you have the same molecular mass, your material is elongated or it is a flattened structure, then certainly spherical structure or shape will settle down faster or will move faster under the influence of the centrifugal field rather than a flattened or elongated structure. So, shape will also play a role, and then density of the particle. denser the particle, more faster it will move in the liquid medium when during the centrifugation. So, these factors, that is size, shape and density, will play an important role as far as particle is concerned when it is put in under a centrifugal field. Now, other factors which are going to play an important role is the medium in which they are suspended, like density of medium, viscosity of the medium, all these things will also play an important role, and also the frictional force, because if you remember in electrophoretic techniques and other techniques we have discussed about, when a particle or an analyte moves through certain medium, and if it is a liquid medium, there will be an upthrust and a frictional force will also be generated. All right. So, what is happening here is that main thing is that through the centrifugation you are able to sediment a particular particle or particular say macromolecule. We can take an example of protein faster. Now, and as we were talking about particle size, when we reach to cellular level, these things like cell or say subcellular organelles, and further if you go like ribosomes or protein materials, lipoproteins or nucleic acids, these are very small, or as far as if we compare the sizes, and for settling or for sedimentation, they will require much higher centrifugation force. So, they can only be like, they cannot be settled just on the basis of gravity, they need to be put under a particular centrifugal field for sedimentation. Now, these particles also have characteristic density, they have characteristic shapes, like proteins could be spherical, they could be elongated depending on whether they are what particular function they have, like say structural proteins are mostly elongated, other proteins which are functional proteins or which are like enzymes, like they might have most of the time they will have a spherical or type of structure. So, they will be accordingly, but they have a particular density, and all these substances which could be say subcellular fractions or proteins will be settled at characteristic centrifugal force. Now, centrifugation can be of two types. One is which like this technique could be both analytical as well as preparative. Now, when you have preparative centrifugation, then centrifugation is done for purpose of collecting a particular material after the centrifugation and for another biochemical investigation. Now, when you are doing centrifugation, I will show you that in a little while, but just to make you understand, the centrifuge provides a means for centrifugation, and it has rotors actually. These are different kinds of rotors which we are going to discuss in detail, but just for now here there could be angled rotor or swinging bucket rotor, or there could be other kinds of rotors which these rotors could be placed on a drive shaft of the centrifuge, and the material which needs to be centrifuged is put in the tubes. For example, there is a central drive shaft, and if you are putting say there is a rotor here, supposing this is your rotor which has a area to be kept in here. So, this rotor can be kept in here on a drive shaft, and this contains areas where you can put your tubes actually. So, this will be in circular fashion, and this has an opening, this could be closed in here. When you open this, then you can put your tubes in these slots. They could be mostly these are even number slots, so that you can balance them and rotor is balanced. So, when you rotate this at high speed, then there is a centrifugal force which is outward, and that will lead to the settling of or sedimentation of the particular material. So, preparative centrifugation will result in certain material which could be recovered after the centrifugation, and it could be used for the biochemical applications or investigation. As far as like this could include, like say you would like to purify or to get at the end, say you have a microbial cells you have harvested and you want to get proteins in there, you can lyse the cells and then you can centrifuge and proteins will come in the supernatant. So, that can be done. Next is that you can also separate say different subcellular organelles like say mitochondria, nuclei or lysosomes and lot of like membrane fractions. Likewise, you can also separate at higher speeds protein fractions or ribosomes and other nucleic acids and other fractions. So, as the speed of the centrifugation, or if we just call in terms of centrifugal force or revolutions per minute of the rotor, then as the speed increases, the centrifugal force increases and the particles which are smaller in size. So, if you increase much more the particles which are much smaller in size also can be sedimented. So, you go from you have centrifuges from low speed centrifuges which are very simple bench top centrifuges to very high speed centrifuges like ultra centrifuge, which can go up to like 600 or 700,000 G force. We are going to discuss what is G force. So, in preparative, that is what is that you recover the particular material. While in analytical centrifugation, what is done, it is mostly you are sedimenting or you are putting in mostly purified or partially purified material for analysis, and analysis could involve say sedimentation behavior of particular analyte, or you want to calculate sedimentation coefficient or interactions, and lot of other studies could be done. Now, when you are doing it, the rotors and there is a detection system which is available in analytical centrifugation techniques or analytical centrifuges, where in real time you can monitor the whole process as it is going on. So, analytical centrifugations are different than the preparative centrifugation. So, these are now if you consider different types of centrifuges, like I said, that could be on basis of speed and capacity, they could be differentiated, like they could be low speed and low capacity ones, they could be low speed and high capacity centrifuges, and they could go up to ultra centrifuges, which is like very high speeds or very high centrifugal forces available in there. There could be these centrifuges could also be differentiated on basis of there are zonal rotors, there are simple rotors which are for small capacities, there are rotors which are high speed and which can sustain high speed and which can have high capacity as well. So, there are a lot of differentiation which we are going to discuss about in terms of types of centrifuges, types of rotors, and how do you maintain those rotors, like taking care of those rotors for their increasing their age or for better performance. Now, another important thing is what we are going to discuss about is certain methods in centrifugation, particularly ultra centrifugation, like density gradient centrifugation or differential centrifugation, and other we will be discussing about certain zonal rotors also, where a large amount, like there are both, one is batch type rotors, another is continuous type rotors. Continuous type rotors is that you keep on applying the sample and also the cells are sedimented, but rest of the material is taken out, and when this is utilized, when large volume of samples are available. Like I said, there could be many liters of volume of sample, but containing very little number of cells, where we will be utilizing continuous flow centrifuges. All right. So, this was little bit like an overview like what we are going to discuss in this section. So, as we were discussing, theoretical basis of centrifugation technique is the effect of gravity on particles. So, like I told you, like particles when they are just suspended in a liquid medium and left, then they will settle down after a certain period of time depending on what is the size of the particle. And this could be like when you are settling them, two particles which are of different mass will settle in a tube at different rates in response to gravity. So, if you want to settle them fast, then centrifugal force is used to increase this settling rate, and this centrifugal force is in terms of multiple of gravitation force. That is, if you say like you can say that this many time of g force or gravity. Now, a centrifuge what it does is it utilizes this centrifugal force or g force to isolate suspended particle from their surrounding medium on either this could be a batch or like I was saying continuous flow basis depending on your application. Now, so this technique in particular is based upon behavior of particles in applied centrifugal field. That is, particles differ in shape, size or density, and on this basis on their physical properties they can be separated as the sediment at different rates from each other in the centrifugal field. Now, the rate is directly proportional to the applied centrifugal field. So, in very simple terms, that if you have two particles, if I say shape, if you have two particles of the same size, but different shapes, they could be separated, because they will settle down according to their shape also. Likewise, if you have two particles of same size, but they have different kinds of like say density, then they could be separated. Likewise, if there are same density particles, but they have different sizes, then also they could be separated. So, these properties could be utilized to separate different kinds of substances or particles from each other and in a centrifuge, and the rate at which they will be sedimenting will be directly proportional to the applied centrifugal field. Also, one important thing that will come up is that centrifugal field of force is outward in direction, and it will also depend on the distance the particle present, the distance between the drive shaft or the central drive shaft and the particle, that is radius you can say. More will be the radius of the particle position from the center, more will be the centrifugal force. All right. So, what is done essentially in this, if you see this figure, there is a centrifuge, a very typical centrifuge, it is a bench top centrifuge, and if you can see here there are like essentially what I want to like to show you is there is a rotor in here inside, and these are tubes which are put in. To enlarge this here, what is done is this rotor which is placed in on the central drive shaft of the centrifuge, which contains a motor to run this. The material or the particle is suspended in a suitable liquid medium, and then this particular medium with particle is kept in these centrifugation tubes. Now, these could be plastic tubes or they could be other glass tubes also, but glass tubes can crack when you are like applying larger centrifugal force. So, now if you see here, these tubes are kept in these slots which are available in the rotor, and this is the rotor screw here which can be closed here, and this is the central position where in the centrifuge it is placed. Now, remember these are centrifuge tubes has to be balanced. So, there are even number of slots, and even if you want to put only one sample, you have to balance it with the other tube of the same weight actually, because otherwise the balance of the centrifuge will be not proper and it will not run properly. So, this is how centrifugation will be done in a centrifuge, in a typical centrifuge. Now, this centrifuge could be very small, like micro centrifuge, where only micro they are like very small amount of samples could be loaded, and could be as big as floor models, which could take many liters of the sample. So, but they will essentially main things are this: they have all controls. There are refrigeration systems, very like ultracentrifuges as vacuum systems. They have means to set up the speed limits, and according to the rotor you can set the amount of centrifugal force to be applied. Remember, these rotors like they specify the kind of centrifugal force which can be applied on them, because the material which they are made of can only sustain certain amount of centrifugal force or certain amount of stress. So, these are all automated centrifuges, and function well, and they are also refrigerated for say biological molecules which can go bad as heat generation might take place. All right. So, the centrifugation is utilized in many different fields, like in the field of proteomics it has been quite utilized. It plays a vital role, and a lot of other fields also, cell biology like molecular biology, it plays a very important role. It has come up like in proteomics, where you require to isolate proteins, and like I was telling you that large amount of harvested cells are taken, where you have cloned and you are expressing a protein, or from cell you are trying to read the proteome of a particular cell. So, where it is necessary to disrupt or lyse the cells, and this could be done by a lot of different methods like sonication or French press or freezing and other methods. Once the cells have been opened up, then all of their contents, that is cell membranes, RNA, DNA, protein, organelles will be mixed in the solvent with and the proteins as well, and then you need to perform centrifugation so that it could be differential or density gradient, as we are going to discuss in later part of this section. And then what is done is that they can be separated out from the non-protein material. Proteins could be separated out, and this could be then within this centrifuge, samples are spun at very high speeds, and the resulting force causes particles to separate, settle down, or pellet actually we can say, and like, and based on their densities and other factors. And as far as pelleting like is concerned, like let me show you what does that mean actually. So, you have say tubes here, and say supposing this is your rotor, and now when these are centrifuged, then you have material evenly distributed all over, like cells, or like say if you have lysed the cells, then both parts of cells like cell membranes, inside content, everything is in here. It is a hazy mixture. Or now when you centrifuge at a particular centrifugal field, what you get is that a pellet and a supernatant is obtained. So, what is pellet? If I say here, then after the centrifugation is complete, then there will be certain material which will be settled on here, on the wall of the tube, and rest of the material which is liquid material, clear liquid material, that is supernatant. So, this is supernatant and this is pellet actually. Now, remember, depending on the kind of the rotor, this pellet, like for an angle rotor, this is angle rotor, they will settle down at this side. If it is a swinging bucket rotor, where these rotors are spun like when they are the centrifuge is switched on, then they will go out, because they are free actually, they are not constrained like in here this rotor, this tube is constrained in the slot, but these are not constrained, they are open. So, they just fly open, and their pellet will be at the bottom, because centrifugal force is in this direction. Here centrifugal force is in this direction, therefore, the pelleting will take place on this wall. Likewise, in vertical tube rotors it will happen. So, this is just to explain that when you are doing proteomics and you are trying to read proteome and you want to separate lot of proteins and you have lysed the cells, then they could be separated out, like protein and non-protein material could be separated on centrifugation. So, there are a lot of applications of this particular method or technique, and centrifugation is capable of doing lot of different things in various areas and for various applications, and all the labs routinely used centrifugation methods. Centrifugation, mostly like every lab will at least have a tabletop centrifuge with a decent capacity, and most of the time they might be refrigerated as well. So, centrifugation is capable of like for example, it is capable of removing cells or other suspended particles from their surrounding milieu on either a batch or a continuous flow basis. Like batch, we have seen just now, like how lysed cell material could be separated from protein solution. So, it could be utilized for isolating viruses and various macromolecules, including DNA, RNA, proteins, lipids, lipoproteins. As well, they could be also utilized for establishing physical parameters of these particles from their observed behavior during centrifugation, that is like sedimentation coefficient, how fast they centrifuge. In terms of Swedberg unit, we will be talking about that. They could be utilized for separating dispersed tissues, the various subcellular organelles, for example, nuclei, mitochondria, chloroplast, Golgi bodies, lysosomes, peroxisomes, membranes, endoplasmic reticulum, ribosomal subunits. All these things could be separated by various methods, like say density gradient centrifugation or differential centrifugation. So, there are a lot of different applications of the centrifugation technique actually, and like I said, it could be both preparative as well as analytical. Now, development of centrifugation, if we go in a bit of history, it started after 1850, and a few like important events which happened during the development of centrifugation. In 1864, Antonin Prandtl invented the first dairy centrifuge in order to separate cream from milk. So, that happened in 1864. Then the potential of the centrifuge in the laboratory setting was first exploited by Frederick Mischer in 1869, and what he did was he used a crude centrifuge system to isolate a cell organelle, and this process led to the discovery of an important new class of biological constituents, and these are macromolecules known as nucleic acids. In 1879, Gustav D. Level demonstrated the first continuous centrifugal separation, making its commercial application feasible. So, the continuous centrifugal separator was invented. Then in 1926, Theodor Swedberg, one of the person who has done a lot in the field of centrifugation, and he received a Nobel also for the invention of the analytical ultracentrifuge, and this was capable of achieving very high G force, like 900,000 G force. When we say G force, it is a multiple of G actually, that is gravitational force. So, he was awarded Nobel for this particular thing, that is invention of analytical ultracentrifuge and his work on colloidal chemistry. And this started a revolution in the understanding of the structure of proteins, like you can analyze proteins with different properties of the proteins in terms of sedimentation coefficient and other in here. Now, French physicist Émilie Henriot developed the first preparative ultracentrifuge, which was able to achieve very high rotational speeds by means of bearing-less top driven and supported by compressed air. So, it was like very high rotational speeds for preparative ultracentrifuge were achieved in very special centrifuges. Then interest in the isolation of viruses brought Edward Pickles and Johannes Bauer together to build the first high speed vacuum centrifuge suitable for the study of filterable viruses. And later Pickles went on to develop the more conventional electrically driven ultra centrifuge. So, there are a whole lot of development which took place in 1900 or 20th century. During the early 1930s, Martin Behrens developed improved centrifugation technique using density gradients of non-aqueous solvents for the separation of nuclei. In 1942, Albert Cloud and James Potter published a landmark paper, that is isolation of chromatin threads from the resting nucleus of leukemic cells. And this paper outlined a series of centrifugation steps in which either the supernatant or the sediment was collected until chromatin threads were retrieved from the final sediment. In 1949, Spinko introduced the model L, the first preparative ultracentrifuge to reach a maximum speed of 40,000 rpm, and this marked the dramatic change in the this particular company, that is Spinko. In 50s, like in early 50s, this saw the introduction of density gradient centrifugation for tissue fractionation, a process developed by a plant virologist Myron K. Brack, working at the Brooklyn Botanic Garden. In 1954, Beckman Instruments, now Beckman Coulter actually, started improving the design of centrifuges, many of which are still used today, and the drive systems were replaced with high speed motors, and the material used for the rotor blade was upgraded. So, Beckman made very popular centrifuges. In 1962, there was like a company based in Hamburg, Germany, and now it is known as Eppendorf, developed the first microcentrifuge for simple laboratory use. In 1976, the world's first microprocessor control centrifuge was launched by Hattich. So, during the 1980s, Beckman launched the first floor ultracentrifuges, and during 1990s, Beckman launched the Avanti high performance centrifuge, which went on to be one of the most popular centrifuges models in the history. Also during this decade, the first centrifuge capable of robotic operation was developed by Hattich. So, this centrifuge also offered PC control and adjustable rotor positioning. There were a lot of other developments where like in 1992, the PAC pressure added centrifugation system was developed by Henkel, and a lot of other development took place. Like in 2000, Eppendorf innovated its line of classic microcentrifuges with the cooled, these were cooled centrifuges and the smallest and quietest you can say centrifuges. And also Eppendorf also launched personal centrifuges like mini spins and mini spin plus. So, there were a lot of development as the time passed, and now the modern centrifuges are very advanced and automated also with lot of options. In future, like now centrifuges have lot of different advanced technologies. Like for example, in case of rotors, rotors have very high tensile strength. The tensile steel is is replaced by materials such as aluminum alloys and particularly titanium in order to withstand high centrifugal force. Most of the high speed rotors are made of from titanium, and they could withstand very high stress actually. There are standard features now included in the centrifuges for say cooling, for programming a particular run, like automatic imbalance detection. Like for example, if you are putting in two tubes, like I told you, you have to balance them. If the balance is not there, the centrifuge will not run. So, those kinds of things are incorporated. Noise reduction has been a very important part of this development, and changeable rotor systems, like you can put many rotors on the same centrifuge. You can put like say Eppendorf rotor or microcentrifuge rotor, or you can put 50 ml tubes rotor, or say swinging bucket rotor. Many different types of rotors are put. So, the centrifuges which are now operating and which are being developed are much more advanced. In very high speed centrifuges like ultra centrifuges, vacuum systems has been added in these modern centrifuges to reduce the friction and maintain the temperature control. So, a lot of automation, lot of advancement has taken place and is still going on in the area of centrifugation, and very good centrifuges are now available for various applications. Till now I have given you an overview of the centrifugation technique. Now, let us discuss about the basic principle of centrifugation. Now, centrifugation method is based on the principle of sedimentation, and when a particle sediments, what it does is it displaces some of the solution in which it is suspended. So, that will result in an upthrust on the particle, and that will be equal to the weight of the liquid displaced. Now, rate of separation in a suspension of particle by way of gravitational force mainly depends on the particle's size and density. So, size and density are the main, and including shape are the main factors which will determine the sedimentation behavior of a particular particle. Now, particles of higher density or say larger size typically travel at a faster rate, and at some point will be separated from particles less dense or smaller. So, smaller particles will travel at a smaller, like they will take more time to settle down as compared to larger or higher density particles, which will settle down faster. Extremely small particles will not settle down or settle out of solution unless they are subjected to a very high centrifugal force. So, like I was discussing in the very beginning, that as the particle size becomes smaller, then the amount of centrifugal force which needs to be applied becomes larger. That is how it will be, that smaller particles will not settle out easily and they require much higher force. For example, if you have to settle down say or sediment certain protein material, then you have to apply much larger force as compared to the, if you want to settle or say subcellular fraction has to be sedimented, where you require a less force. So, when a suspension is rotated at a certain number of revolutions per minute, now remember here there are two ways you can understand this centrifugal force application. One is in terms of revolutions per minute, and another is centrifugal force, which is in real it being experienced by a particle. So, when a suspension is rotated at a certain number of revolutions per minute, then the centrifugal force causes the particle to move rapidly away from the axis of rotation. And another thing is where the particle is present, whether it is very close to the center of rotation or it is far from the center of rotation, both of these particles will experience different centrifugal force. So, the rate of sedimentation depends on the applied centrifugal field, and that is represented if we say in capital G here. which is being directed radially outwards actually. So, centrifugal force, anything which moves in circular motion, it will experience a outward centrifugal force. There is a centripetal force which is inward, and there is a centrifugal force which is outward. And this outward force is given by omega square r. This is which is like it is a angular velocity, and this is square of angular velocity, and r is the radial distance in centimeter, and angular velocity is in radian per second. It is in radians. So, r is the radial distance of the particle from the axis of rotation. So, that is how a particular centrifugal field is being experienced by a particle, and rate of sedimentation will depend on that particular centrifugal field. Now, this angular velocity can also be expressed in terms of the revolutions per minute, and you can write that angular velocity or omega equals 2 pi revolutions per minute divided by 60, or it could be written in terms of g force or centrifugal field, which is 4 pi square revolutions per minute and to r divided by 3600, that factor 60, the square of the 60 here. So, if you see in this figure here, this is typical like direction of the rotation, like when you switch on the centrifuge, then rotor will centrifuge in a particular direction, and there will be an applied centrifugal force which will be in outward direction. Like if it is the axis of rotation, the outward force will be in perpendicular to that axis of rotation, and that will be outward centrifugal force will be applied depending on what kind of rotor, like I said, but the direction of applied centrifugal field will be the same here. As we were talking about the radial distance from the center, if you can see here, if the tube is an angular rotor, as we will discuss in coming time, there will be two, like r minimum, which is the meniscus here from the tube from the center of the rotation, and there is r maximum, which is the bottom of the tube from the center of rotation. So, many times r average could be calculated or taken, as we will discuss later. So, this particular thing could be, these are centrifugal effects you can say when this revolutions per minute are being taken here, or you can take it in terms of angular velocity. So, now G or the centrifugal field is generally expressed like I said at multiples of Earth's gravitational field. So, Earth's gravitational field is all the time present, which is 981 centimeter per second square per square second, and it is the ratio of the weight of the molecule in centrifugal field to the weight of the same particle when acted upon by the gravity alone. It is referred to as the relative centrifugal force or the G force, and the relative centrifugal force can be given by this particular equation, which is like you have divided it by 981 or the gravitational force also to give the relative centrifugal force, and in simplified terms it comes out to be 1.118 into 10 raised to power minus 5 revolutions per minute square into the radial distance. So, this is like how you can calculate relative centrifugal force in terms of the gravitational force, and which is the most widely used term here. So, it could be either revolutions per minute could be utilized, or the relative centrifugal force could be utilized. So, we will stop here in this lecture, and in this lecture we have discussed about the basic features of centrifuge, an overview of the centrifugation technique, where how a particular property, that is size, shape and density of a particle can be utilized to separate them in the centrifugal field, and for this purpose, particular purpose, there are different kinds of centrifuges are used, and as we have seen in the history of the development of centrifuges, they have developed from a very simple centrifuges to very complex and very advanced centrifuges now, There could be different types of centrifuges, right from simple small capacity bench top centrifuges to ultra centrifuges. So, we will continue our discussion in the next lecture, and we will be discussing about we will extend our discussion on basic principle of centrifugation. Thank you.